I will explain you the last one. Okay, so this is the P53 signaling cascade. This is a central pathway which help our cells not to proceed, not to become cancerous. What happens when, if there is any DNA damage occurs, if there is any DNA damage can be there because of stress, because of physical stress, because of mismatch repairs, because of DNA repair mechanism gone mad, because of the absence of a tumor suppressor, because of the presence of an oncogene. There are a lot of hell of the reasons. Because of the radiation, if a DNA damage occurs, remember, it's a normal activity. DNA damage occur every now and then. So our cells, there is what we call as the guardian of the genome. The guardian of the genome, P53. Their job is to keep maintaining, is to maintain the integrity of the genome. That our genome should not accumulate mutations. But if a DNA damage is there, our cells activate P53. Once the P53 is activated, the first thing the P53 is doing, the P53 activate another protein called P21. Remember, it is a cell cycle checkpoint. A cell cycle is made up of different steps. You know, there's a G1 to S phase, from S phase to G2, from G2 to M phase. There are certain checkpoints are present between G2 and S. They are called as G2S. They are called G1s checkpoints. They are called G1s checkpoints. If a cell feels everything is nice and fine, the cell proceed and commit itself to go for the cell division. It start doing the process of replication. If something is wrong here, the cell halts its process of cell cycle. The cell do not proceed for the cell division. There is another checkpoint present between G2 and M, and that is called as G2M, cell cycle checkpoint. It is controlled by other factors. So the P21 is one of the controller of the cell cycle checkpoint called G1s. So P21 can help the cells to feel whether I should proceed or commit to go for the cell division. I should proceed, commit, and go for the DNA synthesis, means DNA replication. If everything is nice and fine, the cell proceed with the cell cycle. But if the P53 is there, the P53 activate. It will block, it will stop the cell cycle. It will stop the cell cycle at G1s cell. But we call it G1s cell cycle arrest. It will not allow the cells to enter into the DNA replication phase. Means cell will not going to commit the cell division. So once the P53 got activated, it activates the P21, and P21 induces the cell cycle arrest at the G1s phase. Why it is required? Because the cells do not want whatever bad is happening with them, that badness, that weirdness, that mutation should not be passed on to the next generation. How the cell can stop it? The first thing that the cell can do, stop the cell cycle. So whatever bad is happening with me, it should stay with me. And that's a very brilliant activity. And that actually helps the cells not, because if there is an error in the genome and the cell committed for the cell division, this error will be passed on to the next generation. And that might become cancerous. So if the P21 is not there, if that machinery is not working, you can guess the errors will be passed on to the next generation. And that exactly happened during the cancer. But this is the normal thing. After that, the P53 is trying to activate the DNA repair pathway or the DNA repair mechanism. It's not here. And the DNA repair mechanism will try to fix the errors, will try to fix the mutations in the meanwhile, while stopping the cell cycle. P53 activate this balance. There is a balance between these two proteins. I want you guys to study this balance between BAX and BCL2. That balance defines whether the cell will going to execute apoptosis or not. This is like a death balance, the balance between life and death. I want you to study which thing will trigger the death. So P53 activate the BAX, and that BAX, if the balance is out, that balance changes the permeability of the walls of the mitochondria. You know, mitochondria is a double membranous structures. Biology mein padha hoga. It is like the outer membrane, and then there is an inner membrane, and there is a mitochondrial matrix present between this thing. The pores of the mitochondria, the permeability of the mitochondria changes, and it releases its matrix. The matrix contains wonderful things, but that also contains a death warrant, and that death warrant is called as cytochrome C. That is called as cytochrome C. The matrix of the mitochondria contains cytochrome C. It is required for the process of respiration, but the bad thing about the cytochrome C is that the cytochrome C is actually an activator of caspases. Caspases are the proteases. Their job is to destroy the proteins. आपने enzymology में पढ़ा होगा, caspases, they are proteases. They can kill the protein structure, they can destroy the protein structure and convert them into simpler polypeptide chains. And caspases are present within the nucleus, within the cytoplasm. खुले आम पाए होते हैं. You might wonder, such a bad thing is present in the cytoplasm. That's very weird, but that is required. But it is present in an inactivated version. But if the boundary of the mitochondria is breached, the cytochrome C leaches out into the cytoplasm and activated the caspases. The caspases will then start destroying whatever comes in front of it. It might be the actin proteins, the cellular structures, the membranes, the proteins present here, the enzymes, all the proteins are now vulnerable. And it is like a bulldozer is like, moving within the building. It destroys one pillar, then the other pillar, then the other proteins, and the cell will start collapsing right from the inside. And that's what we call as apoptosis. So this is, in a nutshell, the P53 pathway. First step, try to stop the cell cycle, so the problem should not pass on to the next generation. And then in the second case, try to fix it. But if the DNA repair mechanism does not able to fix the problem, it will activate the bags, which in turn changes the permeability of mitochondria, releasing cytochrome C and activating the caspases and inducing the apoptosis. It is the death by choice. That is absolutely necessary. So the errors should not be passed on to any other cells. That's the best way to protect cancer. And that is why most of the time we do not develop cancer. Okay. One more thing you should know. This P53 is blocked by the CTCF. A protein is able to block the expression of P53. There's another thing which we call as HOXA10. That HOXA10 is blocked. It's a tumor suppressor. You don't need to remember these things. This HOXA10 is a tumor suppressor. This tumor suppressor can activate P53. And this unfortunate protein, CTCF, is able to block the HOXA10 as well. Remember, in the cell signaling cascade, if the arrows are like this means blocking. If the arrows are like this means activating. So HOXA10 is activating the P53 and CTCF is blocking the HOXA10. I wrote the references here, and you can check the reference 4 and reference 5. Like the other people have discovered this pathway, that your instructor who is teaching you has discovered this thing and this thing in the P53 signaling pathway. So that is why I'm just trying to teach you this slide. Okay, you can see the reference. The two papers are from mine. Okay, let's move to the next slide. Here we go.
Sir, even if the P53 isn't working, can the other cell induce apoptosis?
Please repeat.
If the P53 isn't working, can the other cells surrounding it induce apoptosis?
No, not the other cells. P53 cannot induce apoptosis in the other cells. There are cells which are capable of producing or inducing apoptosis in the other cells. They are called immune cells, natural killer cells, CTLs, cytotoxic T cells. They have the potential to kill the other cells. But in case of P53, is the guardian of the genome. It's like a protector, a savior of the cell. It will try to fix the problem within the cell. If they fail to fix it, it will try to kill the cells right from the inside. So the problem should not pass on to the next. Remember, it's like killing some cells to save the life, because life is about trillions of cells. So kuch cells ki qurbani dena is all fine. Okay, and I have contributed as a researcher in the P53 pathway by finding out that it is activated by the HOXA10 protein, and that HOXA10 is in turn blocked by this protein. So I have found these two information in this pathway and contributed and shared it with the rest of the community in two different research papers. Okay, let's move to the next